Our molecular clock is ticking. We're going to dive into what that means and its implications in the creation-evolution debate, as well as respond to some of the flood of emails we got from the Noah's Flood program. This is Genesis Week. Welcome to this episode of Genesis Week, the weekly program of creationary commentary on news, views, and events pertaining to the origins controversy made possible by the supporters of Core Ottawa, and now carried on Christianema Network, christianema.com, Christian cinema at its finest. A special welcome to our new viewers joining us in the U.S. via the Walk TV network. Excellence in Power Broadcasting, we continue to bring you the information the anti-creationists don't want you to see or hear, and giving glory to our creator while doing it. Remember, if you get lost in cyberspace, you can just punch in Wazooloo.com or GenesisWeek.com and you will find us. And also subscribe to our YouTube channel to get extras like Crevo Rants and full interviews with our guests. I'm your host, Ian Juby. We had a viewer request to dive into the subject of molecular clocks, and hey, we aim to please. So just what is a molecular clock, and how does it relate to creation and evolution? To help us wade through this very technical subject, Dr. Kevin Anderson joins me now via Skype. Dr. Anderson received his PhD in microbiology from Kansas State University, was a National Institutes of Health postdoctoral fellow at the University of Illinois, and later a professor of microbiology at Mississippi State University. He taught molecular genetics and served as the major professor for several doctoral and master's students, as well as research microbiologist for the U.S. Department of Agriculture, and is now present director of the Van Andel Creation Research Center in Chino Valley, Arizona. Dr. Anderson has over 20 papers under his belt in technical microbiology, and has presented papers at multiple national and international scientific meetings. He's also currently the editor-in-chief for the Creation Research Society Quarterly. Welcome to the show, Dr. Anderson.
Thank you. Thank you for having me.
If you don't mind, I'd like to start off with the special request that sort of prompted this interview from one of our viewers. Stephen wrote in from Virginia. Was wondering if you might find the time to do a piece on molecular clocks on Genesis Week. I mention it now because Evos are bringing it to the table more often than they used to in online debates, whereas before they seem to have virtually discarded it. One of the subjects they are bringing it to bear on is plant evolution. One of the worst places for budding evolutionists to start. There's virtually nothing in the fossil record for them to interpret as transition between the major groupings of plants, whereas there is material for them to misinterpret in the animal record. Anyways, an in-depth look at this would be helpful. Now, of course, most of the genetic clocks have nothing to do with plants, but could you please explain to the viewers just what is a molecular clock? Let's start with that.
Yeah, well, I like your statement that in a sense the plants are so unknown that it gives a lot of room for all kinds of wild ideas, and I think that's why molecular clocks are becoming popular in plants, is because you can make all kinds of statements, because genetics in plants is just so strange that anything you say probably can be found somewhere in some data,
Right.
So you get all kinds of room to make all kinds of befuddling statements. But okay, molecular clocks. The basic concept of molecular clock is that under neo-Darwinian principles, under neo-Darwinian thinking, where all organisms share common ancestry, that therefore our genomes come from that common ancestry.
Right.
And so the closer you are to a particular common ancestor, the more that certain parts of your genome should be the same. You all say, if you share a cytochrome gene, for example, with another organism, then that cytochrome from the common ancestor then will have changed over time as evolution's occurred. So when you look at organism A and organism B and you check their cytochrome, then that should be able to give you an idea of just how related they are and how distant they are from their common ancestor.
Okay.
So molecular clock is a way of going in and looking and seeing how much change genetically has occurred in this organism since it evolved from, another particular organism. So
Yeah, and
that's the basic thinking. Yeah.
yeah, and right off the bat, we can see the assumptions of uniformity being tossed in there. The assumption being that these changes that are occurring are occurring and have always been occurring at a constant rate.
Right. And of course, they don't, and that's part of the problem with molecular clocks is that they recognize, well, the changes aren't constant because if nothing else, changes are based upon generation time because you have X number of mutations per generation. So if organism A and organism B have totally different generation times, then that's going to dramatically affect the frequency, or that's going to dramatically affect the accuracy of the genetic clock. And so,
Right.
yeah, the uniformitarian assumption is underlying the whole concept, and yet there's a recognition that it's not uniform. And so you're kind of assuming one thing, recognizing that's not the case, but still assuming it.
Right. Right. And in all of this, too, there seems to be this, again, as you said in your description, this flagrant circular reasoning here. They first claim that we have evolved. They then use the assumptions of who evolved from who. They then try to compare the organisms to figure out when they evolved from each other to figure out how long evolution has been going on for, which is then used as alleged evidence of evolution.
Right. Now, in fairness, if neo-Darwinism was actually true, and I was a neo-Darwinist, I'd be arguing the same thing, because that would be my assumption, is there's no reason to say that if organism A and organism B have a common ancestor 10 million years ago, that I shouldn't be able to then check genetically and see, okay, how much change has occurred in that 10 million years, and then use that to establish a genetic clock. One of the problems, though, is, in a sense, and one of the reasons it becomes a tautology, is I'm assuming, one, common ancestry, and two, I'm assuming a certain time frame. How do I know this particular gene is a better, or molecule, whichever way you're looking, either the sequence or the particular protein, okay? How do I know if it's a sufficient molecular clock or not? What is your calibration to know, does this one work or not? And the calibration is evolution. The calibration is, does it give me the 10 million years that the paleontologists are telling me I need? It does. Therefore, that's the molecular that I can use. That's the molecule. That's the gene that works. If it doesn't, if it says, oh, it really only says 1 million years between organism A and organism B and their common ancestor, obviously that doesn't work. So that molecule, that gene, doesn't work. So my calibration assumes evolution. And we see that just repeat throughout all of the molecular clock work, is you assume evolution, you see which marker works to fit the already preconceived idea of what evolution is, and then if it works, then we say, voila, we've proven evolution.
Sure.
Yeah.
Sure. Okay, now one of those clocks that I'm sure most of the viewers have heard of, certainly I've heard a lot about it, I've even talked about it on the show, we've heard lots about mitochondrial Eve.
Yeah.
And in fact, as I mentioned it on the show. So this is a mitochondrial DNA molecular clock, correct?
Right.
So what's the story there?
Okay, the story is that all eukaryotic cells have a little organelle called a mitochondria. That's, of course, the site where most of the ATP is made. The mitochondria also happens to have a little bit of piece of DNA. Now, most of the time, a little controversy, but most of the time, you get the mitochondrial DNA from the female. So, for example, you and I both have our mom's mitochondrial DNA. She got that from her mom, et cetera, et cetera. Okay, so when you have that kind of lineage characteristic, then that already gives you the ability of being able to follow a specific strand of DNA back through particular family histories. Now, like any piece of DNA, the mitochondrial DNA is subject to mutation. In other words, every once in a while, a change may be introduced into it. Okay, so we can track these mutations and see, all right, how much change has occurred between your mitochondria and your great-great-great-grandma's mitochondria. And so by doing that, say we had in the late 1980s, some geneticists that did a study where they determined two things using mitochondrial DNA. They, one, determined that all contemporary humans have the mitochondrial DNA from one female, and that's why it became known as mitochondrial Eve.
Sure.
You know, and they named this female Eve not out of any kind of deference for even the Bible. It was a joke. It was tongue-in-cheek, making fun of.
Yep.
But of course, the fun thing of that is now they've introduced Eve into the scientific literature, so we can go into the public system and talk about Eve because it's in the public, it's in the public literature, it's in the scientific literature. So,
that's right.
God has a way of always working these things out. So it's kind of amusing.
Yep.
So anyway, they identified what they determined was this single female, identified her as mitochondrial Eve. Now, they weren't saying she was the only female alive at that time. They're just simply saying that she's the only one whose mitochondrial DNA has survived up to this point.
Right.
Everyone else, all the other females that were contemporary of hers, their mitochondrial DNA has died out. It hasn't been passed on. Okay, so that was one thing they found. But it is interesting how that is totally consistent with the biblical Eve, mother of all nations, mother of all humans.
Yep.
The second thing they did then was, as a typical molecular clock, they worked to determine, okay, just how long ago did this Eve live? In other words, we can take and genetically determine what's the distance between us and her in terms of years. Now, as the old saying goes, an interesting thing happened on the way to the forum. An interesting thing happened on the way to publishing.
Yep.
When they calculated mutation rate, they found out that it didn't give them the number that they wanted.
Right.
In fact, there's a paper in Science in 1998, almost 10 years after the publication of the original mitochondrial Eve work, where there was the admission that if you use mitochondrial DNA based strictly upon its known mutation rate, in other words, on the data, you get an age for Eve of about 6,000 years.
How very convenient.
Of course, that is completely unacceptable. There's no way that's going to work, and no way you're going to get that published in the scientific literature. That would go over, literally the proverbial lead balloon.
Yes, yes.
So, enter in evolutionary thinking. See, because we got to think like an evolutionist, not a scientist. Think like an evolutionist. Okay. Evolutionists would think, all right, if the current rate of mutational change, the uniformitarian idea, is not working, it's not giving us the right age because it's giving us 6,000 years, and we know 6,000 years can't possibly be right, okay, then there must be a fudge factor necessary. So the fudge factor was, they calculated, okay, chimpanzees and humans have a common ancestor 6 million years ago. I think since then it's moved to 5 million years, but why quibble over a million years? Anyway, okay, so 6 million years ago. So what they did was they took the mitochondrial sequence of the chimpanzee and the mitochondrial sequence of the human, and they compared those and then extrapolated 6 million years and determined what kind of change that brought on.
Right.
And then they calculated mitochondrial Eve's age. See, and when you do that, then you get the reported 150,000 plus or minus years.
Right.
That's where that time came from. It has that evolutionary fudge factor in there. In other words, back to the tautology. If we assume evolution and we bring evolution into our calculation, in that evolution then gives us a fudge factor, so we can fudge the data to make it fit what we want, then voila, it works. We make it
voila.
What we want. And therefore, mitochondrial Eve lived 150,000 years ago.
Right.
If we go with the real data, what is the actual mutation rate? Well, it's only 6,000 years. Okay, well, obviously that can't be right. And so that's one of the things fascinating about mitochondrial DNA is that it supports very strongly the biblical view of Eve. Also, that's why you don't see very much written about mitochondrial Eve as a molecular clock anymore, because they lost it.
It doesn't work. Yeah.
And so more or less it was, well, obviously, and that's one of the things that the 98 paper in Science concluded. You can't use mitochondrial Eve as a molecular mark, as a molecular clock, because it doesn't work. Why doesn't it work? Because it gives us 6,000 years. So it obviously cannot work. See, what is our criteria for determining the accuracy of our clock? Does it work for evolution? Mitochondrial Eve did not work for evolution, so therefore the criteria says it must not work.
Right. And change the criteria then to make it work. Okay.
Yeah.
And now, of course, this may be a little too long to get into, but of course there is the Y chromosome atom, as they called it, which was similar to the mitochondrial Eve, but just looking at the first man.
we guys didn't want to be left out. Exactly. Because you get your mitochondrial DNA most of the time from your mother.
Right.
You get Y chromosome obviously from the father. I mean, that there is no doubt about. There's no controversy in that. You have to get it from the father. So the idea is, okay, well, it works so well for mitochondrial Eve and for the women, to determine the mother of all people, let's use the Y chromosome and do the same thing as a molecular marker. Let's track the changes that have occurred in the Y chromosome through the males.
Right.
Now, an interesting thing again happened on the way to the forum. Just keep not getting there. What happens? Okay, we're looking at the changes that occurred in Y chromosome. The problem was that there wasn't enough changes that occurred. They couldn't find adequate changes. So the mutation rate was way too low to calculate. And therefore, because it was way too low to calculate, they did a very good job of saying that Y chromosome Adam, is 280,000 years plus or minus 280,000 years.
Plus or minus what?
Plus or minus 280,000 years. In other words, because they couldn't find sufficient change in the Y chromosome, they just made up a number, 280,000 years.
Okay.
Oh, yeah. And one of the things they state in the paper is that they had to have a date that they felt was consistent with Eve.
Right.
I don't know why. I don't know why that was so worrying about them, but they had to have a date consistent with Eve. So 200, I guess, just out of thin air, 280,000 years. They don't explain where it comes from. It's just 280,000 years plus or minus 280,000 years.
Excellent.
Yeah. I wish I could have those reviewers. Man, what great work. Now, again, in fairness, there has been more detailed work done with Y chromosome in the last 15 years, and he's gotten younger. Y chromosome is younger now. Now he's not 6,000 years. I haven't gotten it down to that point yet. But last I saw, I think he was about 40 to 60,000 years old. So he keeps getting younger. Okay? But again, nothing has changed as far as the data is still not very accurate, as far as there's just not enough change occurring in the human Y chromosome to allow us to be able to determine a molecular clock very accurately to give us a Y chromosome atom. So they still continue to suggest then that obviously Y chromosome atom had to live Y tens of thousands of years ago, because that's what our evolutionary paradigm suggests.
Right.
The real data is we can't calculate.
Okay.
We can't calculate Y chromosome atom. It's just as likely as yesterday.
Right. Okay.
But you don't get that published. So
right, exactly. Yeah, who would publish that? Yeah, we don't know.
They just made up that fit with what they wanted, and that's how they get it published.
I see. Okay. Yeah. So now, speaking of molecular clocks, we know the decay rates of DNA, the deterioration of flesh, soft tissues, etc. And years before Dr. Schweitzer found soft tissues in a T-Rex bone, the Creation Research Society published soft tissue.
In 2001, Mark Armitage had reported the detection of collagen in a T-Rex fossil. Right. And that was 4 years before Dr. Schweitzer's first publication. But if we look in the literature, interestingly enough, we find that even Mark wasn't the first one. Other people reported it too. It's just Mary's is the one that's received the most attention. But it's been in the literature for a number of years. But they didn't make a big deal out of it. It was like, okay, this would appear to be, this could be, this might be. It does look like this is consistent with, they always hedge it with those kind of words. But we've seen then for quite a few years that there has been reference to what appeared to be soft tissue and what appeared to be proteins in dinosaur fossils.
Yes. Yeah. And of course, just recently, I'm assuming you caught this paper again. Dr. Schweitzer, just 1 or 2 months ago, finding now DNA in a hadrosaur bone.
Certainly finding evidence of. I guess the best way to put it is she used an antibody that had a binding affinity for certain regions on DNA.
Right.
And antibodies found binding sites. Even though she hasn't actually specifically chemically detected nucleotides.
Right.
She's detected molecules that these specific antibodies are binding to.
Right.
And she also used a stain that has a similar specificity.
Right.
And so she has been able to indirectly detect DNA by these antibodies and these stains finding sites to bind to.
And of course, they react with DNA. That's the whole point.
Right,
Okay.
right. And so that certainly is a precursor to that. There's obviously something there that is reacting as DNA would react.
Yes. So now my interest here, of course, is Creation Research Society had started up what they called the iDinosaur Project, and I've sort of followed along with this. So what can you tell us about that?
You can go to our website, which would be www.creationresearch.org, and we do have on that website a somewhat detailed description of what we're doing, what we're trying to accomplish. Part of it really comes down to is we're not in any way questioning a lot of Dr. Schweitzer's work. In fact, she's really done some very good work. I know she gets upset when creationists talk about her work. But
she has. She's done excellent work.
Because it's certainly totally consistent with what we've been saying. And so how is it unfair for us to take data that's totally consistent with our position and use it? Where does that come from? She's accused us of rewriting the rules. What rule is it that we can't use data that's consistent with our viewpoint? Our viewpoint is dinosaurs did not live 68 million years ago. And the fossils that are digging up were not 68, 70, 85 million years.
Right.
The presence of the tissue, the presence of the proteins, the presence potentially even now of DNA is consistent fully with that they're not 68, 70, 80 million years old, that they're just a few thousand years old. So why wouldn't we talk about that? I mean, it would be unscientific not to. So in that sense, regardless of what evolutionists may want to think are or not the rules, of course we can talk about it. Yeah, Dr. Schweitzer's done some very good work, but we just felt that it was appropriate for us as a research society and as a research facility in Arizona to obtain our own samples. We have the microscopic capability. We have some of the protein capability. We have some of the DNA capability. So it's appropriate for us to get our own samples to do some of our own work so that we're not dependent upon other labs to report or not report what they find, depending on if they want to or not want to. And so therefore, we're going to become a little bit autonomous and report our own work, report what we want to report as far as what we find, and not be worrying about, is this going to be accepted by the evolutionary establishment? Is this going to get me in trouble at my university if I publish this?
Sure, yeah.
I'm not accusing her of doing that. In no way am I accusing her of doing that. I'm just simply saying that we felt that it was totally appropriate for us to become independent so that we now have control over what is done and what is published.
And it's replication of results anyway. It's proper science. Good science.
Replication, which any scientist should applaud. Let's replicate results. Isn't that part of the empiricism of science? Is that not only do you replicate results in your own lab, but other labs should be able to take and replicate results also.
Right. And
even
so
if we are those easy, wild, nuthead creationists.
Right. So now if people wanted to help out with that project, okay, funding obviously is one thing. Like, are you looking for dinosaur bone samples? I don't know.
We don't get funding from National Science Foundation or the Carnegie Institute or anything like that.
You don't?
Funding is, funding is strictly from individuals, churches, other organizations that support our work and want to help fund our research.
Okay.
So, and like any research, there's always money. I mean, money drives research. Research is expensive.
It is.
It's expensive equipment. It's expensive for time. Expensive for travel. We've had to travel to go collect samples. We pay other people for samples and such. So there's a nice money demand to it.
Okay. Well, you're getting me started now too. I want to go off in different tangents, but we better call this quits. But thank you so much for being on the show today. I know for fact the viewers will really appreciate it. So thank you.
Well, thank you for having me.
This interview was edited for television, but you can catch the entire interview on our YouTube channel. Just head on over to genesisweek.com, which takes you to our YouTube channel. Stick around. We'll be right back after this break that is so short, it won't even show up on your molecular clock. This program sponsored in part by Genesis Park, found at genesispark.com, where we say dinosaurs are living evidence of a powerful creator. And by Canada's first permanent creation museum in the heart of Alberta's dinosaur beds, the Big Valley Creation Science Museum, bvcsm.com. And by you, our financial supporters. What does the Bible say about aliens? Is there life on other planets? What can science tell us about the possibility of aliens? Ian Juby gives answers to these and many more questions in this fascinating and highly disturbing subject. Looking analytically at the subject, complete with the testimonies of people who claim to have been abducted by aliens. The answers will probably surprise you. In this 1.5-hour lecture, Ian shows that the alleged aliens are a problem and that Jesus is the solution. Order online today at Ian's bookstore. Mail for me? As predicted, our Noah's Flood show garnered its own worldwide flood of emails and criticisms. Oddly enough, not one critic actually attempted to refute the evidence I pointed to in the show. You know, water gaps and wind gaps, planation surfaces, quartzite boulders with percussion marks, etc. Instead, the overwhelming response of the critics was one of mocking and saying the ridiculous things like, "There's not enough water to flood the planet." You guys are such kidders. Now you guys know me, I enjoy a good joke, so let's take another look at the Earth, nicknamed the Watery Planet, and please tell me again that there's not enough water on Earth to flood the planet. Yes, claiming there's not enough water is quite a humorous claim that I hear all the time, in spite of the fact that some 70% of the planet is covered in very, very deep water. Every major mountain range on planet Earth has fossilized sea life on it, and the mountains are composed of the layers which fill in the lowlands on the continents, so obviously the lowlands were underwater as well. Then all the layers tilted and buckled up during mountain formation. Yes, even the top of Mount Everest has fossilized clams on top of it. Clams which were buried alive in the closed position. Now take my word for it, clams are lousy mountain climbers. I base that statement on extensive, in-depth scientific studies I conducted. We all saw the horrific events of Indonesia in 2004 and Japan in 2011, when in each instance a very tiny island moved only a few feet. The massive inland flooding that occurred was nothing compared to the global flood, which we would contend happened when entire continents moved, and rapidly, as can be convincingly demonstrated by studies of the oceanic floor fracture zones I discussed in previous shows. So, it is utterly ridiculous to suggest that there's not enough water on Earth to flood the planet. Okay, I gotta call that quits for today. Thanks for watching. I'm your host, Ian Juby. I hope you'll join us again next Genesis Week. Remember, you can send in your feedback in a number of ways, and let us also remember the words of our Creator, the Lord Jesus Christ, who said and warned us, "I am the way, the truth, and the life. No man comes to the Father but through me." We'll see you next week. We need your support to help keep this program on the air. You can help by making a tax-deductible donation to Core Ottawa, Canada North Post Office Box 72075, Ottawa, Ontario, K2K2P4. You can also sign up for Ian's newsletter detailing current research and news items at ianjuby.org.